I want to thank you all for coming, and I'm happy to provide refuge for our Helston and Douglas Lake population, where I understand it's 33 below zero today. So, Mark just grunted. So, in particular, I want to thank the Ann Arbor Biostation staff. Alicia has been working very hard. Peggy, behind the scenes, Carrie, Jason, and Pam Ballard, who's picked up slack while everybody else is planned for this meeting. So thank you all. I want to thank, and I don't know if anyone's here from the college except Todd, but the College of Literature, Science, and Arts has basically funded this meeting. And so I really want to extend a thanks to them or to us. I'm part of the Literature College, but the Dean's Office has supplied money, recognizing the value of what the station brings to the university and to the greater scientific world. So thanks to the College of LS&A. Thanks to Todd Austin, who is running this and making this meeting available as a live cast and also as a recorded meeting online. So my understanding from Alicia is that you can rebroadcast on your laptop our segments of this meeting, so you don't have to watch the whole thing at once. And anyone who's not here, thank you also for tuning in. I just wanted to mention briefly the agenda. The morning talks from just after we finish until 10:15 will be they're selected as overview talks. Themes we'd like to cultivate, develop for the future. And then we have breakout sessions both before and after lunch. I won't list the times because you have agendas in front of you. But those are times when we can actually get together and think creatively and strategically before and after lunch. Then in the afternoon, we have a series of talks describing ongoing research. Of course, that's related to the future, but these are more focused talks on particular projects. In the afternoon, we'll have an information session for faculty and researchers. 3 information sessions. Yes. Oh, Jason, did you want to say something?
5 total. There's 2 more on the other side.
Oh, there's 2 on the other side. Thank you. Okay. And then a reception from 5 to 6. Dinner on your own tonight. And I will not take any more of your time. Tomorrow morning, I'll talk briefly about the state of the station. But I'd like to thank Todd and Todd Austin from LSA Instructional Support Services would like to tell you a few things. So,
good morning. This is just a quick little intro since I have.
I guess that's what you.
That's okay. Hold on to it. I'll just project.
Okay.
All right. Keep it simple here. Just a couple of things about the technology being used this morning and its availability. I don't know how many of you are LS&A. I work for LS&A. Many other units do something similar, but not everybody is aware of BlueJeans. It's been available for about 9 months or so. It's a video conferencing cloud service. Anybody who gets a paycheck from the university can get access to BlueJeans. You need this sort of access, which you get up there, just with your UMBS credentials, umich.bluejeans.com, only to create the meeting. Nobody needs an account to join a meeting. So our distant folks who are joining today are not using any kind of account. All they need is the URL. It is a cloud service, so there are dedicated BlueJeans servers in the sky that connect all this. They interpret between all these different kinds of technology. The web browser, there's a browser plugin. There are mobile apps for iOS and Android. It'll talk to a plain old telephone, and it talks to a video conferencing unit like I have over here that we take around all over campus and beyond for meetings. It does support up to 100 different points in the same meeting, so you can have a fairly large meeting. It has significant advantages over Skype and Hangouts. One is that we can use a unit like this in a classroom or a meeting room, which is vastly superior than trying to use somebody's laptop to support the conversation. Skype does what's called peer-to-peer networking, so it uses the computers of logged-in Skype users as routers rather than having Skype service in the sky. And it means that your call suffers when any of those computers has any kind of problem, which doesn't happen with BlueJeans. And of course, you can record with BlueJeans. So we're recording right now. Turns out a nice 720p MP4 video file, about 500 megs an hour. So it's pretty chunky, pretty good quality. Lets you do what you want with it. As I say, up to 100, so much better than Hangouts. I found that Hangouts tend to degrade when you get beyond about 4 or 5 people, and Skype, our BlueJeans doesn't suffer from that. So what do we do with this BlueJeans tool? We do lots of meetings like this. This is one of our larger meetings, but we do lots of smaller meetings too. That includes things like dissertation defenses and faculty meetings, job talks. Some faculty members distant, can't make it to the talk, we bring them in. We do classes a lot. So guest speakers, instead of your laptop and Skype, you can do this. But we also do term length classes that are shared with other universities, but maybe just a professor here, a professor someplace else, but we also knit together universities where classes are co-taught at each end. And they may both be American universities, but we do a lot of foreign partner things as well. And if you have any thoughts in those areas or any of these areas, I urge you to come and speak with me. Interviews, job interviews, things like that, conferences like this one, connect people to, dissertation defenses on which, they're a committee member and the dissertation defense is someplace else. HIPAA compliance is not quite there yet. It's merely a paperwork matter. If you have personal identifiable information that you need to protect, working on the documents that have to be signed between you, and BlueJeans, technically it's all there, but legally it's not quite there yet. And a couple things to remember. If you have a request for service or want any more information, email us at this address. It goes to my group, and we will be very happy to assist you. We ask for 2 weeks of notice if you can give it. I've done it on 10 minutes notice, quite frankly, but you can't always count on us being available when you do that. And if you have a class or something like this, it's full service. We come in and set it up, and we tear it down and take it away. During a class, we'll run it, so we do pan tilt zoom on this camera and pick out your students, and they all are full face or close to it on the screen for your distant guest. But we also have self-serve options. We loan out a couple of different Logitech webcams, very nice USB webcams, a smaller one for a group of maybe 5 or 10 people, and a larger one that's better quality that might do about 20 people. They should just run off a computer. Any quick questions? Yes.
Would you be willing to bring this up to the biological station?
I think there are tremendous possibilities with the biological station. I'm really happy to be here for that particular reason. I think we should be using it at the biostation. And I'd be really happy to work with you folks on making that possible. I know you have a good, high-quality internet connection up there now. Oh, this is highly forgiving, I should say. I punch in connections to Haiti regularly, rural Haiti, North Korea, Myanmar I've done. So it's very forgiving to poor bandwidth and unstable bandwidth. But yes, that's by all means, I'll be talking with Alicia about that in the future.
Yeah, and Bob Vanderkoppel got on BlueJeans, and he'll be popping in too to the meeting today.
He's online right now.
Okay. Oh yeah.
He's attached. It's just have his camera turned on.
Okay. So yeah. So
okay.
Bob's here.
All right. Thanks very much.
Thank you, Todd.
Thanks a lot, Todd. Okay, what I'd like to do is ask speakers to talk for about 10 minutes and allow 2 or so minutes for comments and feedback. So first speaker is Denny Albert from Oregon State University and the biostation.
Well, thank you. Can you hear me? Yeah. I'm broadcasting here. And you have
Yep, that's it. So, forward, back, laser pointer.
Oh, okay. All right, we'll see how well I can do this laser pointer. Okay, I'm talking, I have 10 minutes. I'm going to try to talk through 35 years of data collection at Colonial Point, so wish me luck. I'm sorry, Colonial Point. That's only 30 years of data. So Cecil Bay is a project that Ed Voss started in 1971, and then I did some further work in Mackinac Bay. And we're talking, the primary change that we see over this 35 years is water level fluctuation. And you can see on the left, that person is sampling in a dry water year. And you can see what it looks like in a high water, high energy year. A lot of difference in the same wetland. Okay. Well, let's. Okay. So Cecil Bay, actually, I'm going to focus most of this on Cecil Bay. And we've got a lot more data there. That's 35 years of data. That's an open embayment that's just to the west of the Mackinac Bridge. You can see it has about 10 miles of open water. So during major storms off of Lake Superior, it can be a pretty rough place to be either a biologist or a plant, and probably any other kind of animal. Mackinac Bay is much more protected. It's sitting up here behind Marquette Island, and Mackinac Bay is right next to Cedarville, which is why I referenced Cedarville. A lot more people know Cedarville than Mackinac Bay. So very protected marsh, and some major differences because of that level of protection, but I'm not going to have time to go into that at all. Here's the kind of difference that we're looking at. You can see in 1998, the water level is dropping, but this is the entire marsh here, where that red arrow is the transect, or you see a red arrow there? Yeah, okay, I can't see it from here. So the entire marsh is pretty much flooded in 1998. It was completely flooded in 1997. 2012, the water level has been down for quite a few years, and the entire marsh that was sampled was dry. There are a lot of questions we can ask here. We can, with the water level fluctuation, we can see how it influences marsh structure. There's some models that I'm not going to have time to go into much detail, but that are going to be probably better explored with this database than any other database that I know of in the Great Lakes. Plant diversity and species richness at multiple scales within the wetland. So you could look at the entire marsh, or you can look at one zone within the marsh, or one plot over 35 years, and some very different dynamics there. You can look at some of the dominant plants, Chenoplectus acutus and Chenoplectus pungens. Many of you may know them as Scirpus acutus and Scirpus americanus, the bulrushes. They're the dominants in the wetland, and they change. Their dynamics are also very interesting. And we can look at the dynamics of cattail, invasive cattail, and Phragmites. So a lot of questions that can be looked at here. The model that a lot of people work with in the Great Lakes, they started with Ketty's model, which looks at water level fluctuations. And then there's Ketty and Resnicek. I didn't reference that. And then Maynard and Wilcox. The drawing here is actually from Maynard and Wilcox. And at the top, when the water levels are high, you get rid of a lot of shrubs and large emergent species. When it drops, all of a sudden you got a seed bank and this tremendous amount of regeneration. If it stays down, then you start getting the shrubs and emergents coming back. And so it's a very dynamic system. But this database is probably the first time you can actually evaluate that adequately. It's one of the best databases to evaluate this model. If you look at the data collection, you'll notice that we collected along quadrats, starting close to the dry edge, close to the road, and we go out to 200 meters. I only showed half of the database, and we record the water depth along there. And this year, you can see there were 8 of the plots that were dry. And then for every species, we record in a meter squared area how many stems occurred. You can see for this species, Agellinus purpuritis, is in the first 25 meters of the transect. A lot of the taxa, we had 160 or so taxa, and you can see there's a lot of these, and I only showed part of them. Step back this way, this way, this way.
Turn off that mic.
How's that? Okay.
That's better.
That's better. I like that. So you can.
Okay. I'll just take this.
Okay. So you can see there are a lot of species here, and most of them only showed up once or twice. There were low numbers of stems, but some of the bulrushes, Scirpus americanus, showed up in 800 out of the 1,300 plots. So 1,300, that's 43 plots by 35 years. It showed up a lot. as a few of these species did, and there were 44,000 stems that got recorded over the period of 35 years. So really great database for looking at some of the dynamics of these common dominants. The bathymetry, very, very flat wetland. Most of our coastal wetlands are. The slope here is 0.36. That means if you drop the water level a meter, 280 meters, 278 meters of the marsh dry down. And the other marsh, Mackinac Bay, is a 0.34 slope, so almost identical in terms of the slope. Very, very dynamic system. This is the water level over this period from 1965 to 2008. It includes the 35 years. And probably the important points here, this is the low water, 176 meters. This is high water, 177.4. So 1.5 meters, that's about as tall as most of us are, I think, maybe a little less. But really dynamic system. And during that period, we had mean high water occurred a couple of times, and we were close to the mean for a period. And then we went into this low water. We may be seeing some global warming or some impact of some of the dredging probably as much from the dredging of the St. Clair. But if you look at this all the way back to 1914, we see the same cycle 3 times. So almost identical 1.5 meter water level fluctuation 3 times occurred since 1914. Trying to look at this entire marsh over a 35-year period is tough, so this is the best I could do. So here's the transect going 200 meters out into the lake. Here's 1971 to 2001. And you'll notice that in the first few years of the transect, the 1970s, the entire marsh was flooded. There was no place dry to stand, and so the plant diversity was low. It was in the range of 11 to 18 species. Over that period, we got into the driest phase. The same thing happened here in the 60s, this long-term dry phase, and we're talking 140 plus species. A single plot can have 40 species in it. What we don't see in this database is the dynamics that occur here. So during the high water, this sedge meadow is being eroded away, and all those organic material that's the seed bank for the next low water period. Same thing's happening out in the bulrush beds. And this is a combination of storm waves and ice. This is the type of changes that you're seeing. So here we're looking at just the meadow zone, and in a high water we have 36 species. In low water we have 88 species. So this is 4 years apart. If we go to Mackinac Bay, we see the same pattern, 23 and 43 species. And each one of these trees, shrubs, grasses, vegetation types shows a similar decline, except for the ferns. They're pretty stable. Now, if we look at a single water depth, here we're looking at 4 meters. The red is the water level fluctuation, and it goes up to 40 centimeters in depth at 40 meters into the transect. The dry part of the transect can have 40 to 50 centimeters of water when the water level is high. Notice the high water period, plant diversity drops to almost nothing. When it's dry, diversity climbs up very rapidly. 1 year, you can have, from 1 year with 4 species, the next year you can have 40 species in this plot. And actually up to almost 50 species in this plot with a drop in water level. Very dynamic system. If you go out to 60 meters, so you're out into the part of the marsh that's wet most of the time, you'll notice that it stays low in diversity the whole time it's flooded. Doesn't matter how deep the water is. And then we have a dry period here, and all of a sudden the water level rises. We can look at the bulrushes, and you can see here, one bulrush is really dominant in the protected part of the marsh. This is dominant in the outer marsh. And some of these relationships are related to soil differences that occur. Now, we have very little soil data. I collected it in 1980 when I did the study. And what we found is in this first 70 meters, 80 meters of the marsh, you have organic material accumulating, sand, gravel. This is the dynamic part of the marsh that gets all of the deposition. And then you go further out, and I only showed a little bit of this part of the marsh, 60 meters, 50 meters of it. This is the zone of erosion, and you're down into the base clay. So this soil aspect is real important in understanding the dynamics, just as the wave energy and ice scour is. Okay, I think I used my time.
Thanks, Kenny.
Test. We're just going to test this again.
Okay, we're testing that, but it does
Check.
comment or two.
Any questions?
Yeah.
Did you get any algal species?
Algal species.
Or anything?
We didn't do any looking at the algal species, but one of the things that's noted, the notes that aren't in the database are almost as are very instructive, and they comment during the high water period in that close to the shoreline that you have these huge algae soup bowls, and so yes, algae are probably important, and that's important probably in the dynamics as the water level drops. They decompose very rapidly, and you get a lot of nutrient availability.
Okay, thanks. Our next speaker is Paul Moore.
Me.
From Bowling Green State University.
Okay.
It's a-
All right, can you hear me?
Stand up and you have about a minute left, like at 9 minutes. So that means you're ready for 1 or 2.
Okay. If Denny can do 35 years in 10 minutes, I can probably do 20, hopefully. Is it advanced? All right, so this is the real talk of the title, and the reason this is real talk, I'll talk to you about that in a second. We work out at the Stream Lab at the station, and if you're not an aquatic ecologist, you probably don't know where the Stream Lab is. It's right about here, so it's about 4 miles from camp straight away. It's on the east branch of the Maple River that takes surface water of Douglas Lake and moves it from Douglas Lake down to Burt Lake. What's nice about this facility is that when we come out in the springtime, this is a concrete pad, and we take about 800,000 gallons of the Maple River, dump it on this pad, do something with it, and let it flow back into the river. When we come out, there's nothing here. We've got water flowing here, but this pad is empty. So we move about 300 to 350 cinder blocks every year, and then at the end of the year, we move them off. So this is the love-hate relationship. We move these cinder blocks because we can construct different stream habitats. So what you see here is the beginning of the year, and we have a series of experiments over here and experiments over here, and I'll get this right. Here we go. And a long channel here. And then over the course of about 12 hours, with all my students and whoever else is out there with us, we go from a flat concrete pad with 1 row of cinder blocks, and then we add another row of cinder blocks, and we put plastic in it. And then once we add the plastic, we put structures on top of it, which is probably not OSHA certified structures, and then we put electricity, so electricity, structures, and water all together. And finally, at some point, we bring in some plumbing here. Probably not up to grade plumbing either, but it works. And we have a stream. So this is about a 60-foot stream here from here to here. And what this allows us to do is change anything we want about the stream. We can change the flow velocity, we can change the temperature, we can change the substrate. That's the experimental part of this facility. When we have streams like this, we can have chambers like this. This is an older experiment where we did elevated ozone, and the impact of elevated ozone on algae and macroinvertebrates. The cinder blocks here are actually a cooling chamber. So this has got cold water that keeps these circulating small-scale streams at a certain temperature for what we need. So the nice thing is, it's like Legoland for scientists. We come out, we imagine what we can do, and we're limited by imagination and how many cinder blocks we have. We can set up a number of different experiments. So this is a lake system. It's about 4 meters by 4 meters. It's a very slow flow system, and we put animals in here and look at animal behavior. This is a long channel, so if we want to simulate a stream and change stream dynamics, we can do that here. Up on the top here, you see tabletops, and this is where Bob and Rex and Pat and algae people come out, and they put a rain gutter here. And we can do things to these rain gutters. We can add pH, we can change temperature, we can do nitrogen and phosphorus and all that kind of good stuff. And we have replication. The nice thing about ecological studies out here is we have true replication as opposed to something else. We have habitat selection here, and this is actually a student project over here. It's hard to see, but it's a meandering stream, so these don't have to be straight. So we can really replicate what is going on in a real-world situation in an experimental setup. So it's really nice and flexible. This is in detail. Sometimes we use the gravity of the water, or the pressure of the water to drive the systems. Other times we have air stones coming in here, and this is made out of PVC pipe. And we can have circulating water that way. So the scale of what we can do is anything from an algal cell up to Paul Webb's been out there with some large-scale plumes doing fish dynamics. So lots of scales, lots of time scales, space scales, so it gives us a lot of flexibility. This is kind of like a commercial Quantum uses. There's lots of space out there. So at the end of the year, we end up with something like this. So instead of an empty concrete pad, we have 300 some cinder blocks. You notice if you were to come out in the beginning of the summer, and you can help us move cinder blocks, you notice giant pads of cinder blocks here, but they're all used. So we have this total facility is full of cinder blocks. So we have a chance to take this and do anything we want. And every year this looks different, except for this long stream here because we got sick of moving these cinder blocks. So everything gets reconfigured year after year after year. So why do I do this? I'm interested in this chemical, or in this question. How do chemicals impact aquatic ecosystems? But everything I've talked about is flow. So why do we have flow? Because if I were to put a pile of chemicals right here and then say, how does it affect this room? It wouldn't. We need flow, whether it's terrestrial or aquatic systems. Chemicals don't impact ecosystems without flow moving that chemical through the environment. So what we do is a combination of ecology, chemical ecology, and hydrodynamics. By controlling the flow, we can control what the chemicals do for that system. It gives us a nice advantage. When I say chemicals, we're really broadly interested in chemicals. I have a whole bunch of different types of chemicals. So we have the standard nitrogen and phosphorus. So if we want to do those kind of additions and see how that changes algal community structure, we can do that. We're doing work on genetically modified organisms, particularly BT corn, or other types of crops that have stacked herbicides or pesticides, and what happens is you grow those up, and then you harvest them, and those chemicals get into the streams that surround those agricultural fields. Well, those chemicals in here, particularly BT, can target insects and kill them, but they also have non-target effects. So we look at that kind of stuff here. This is kind of hard to see. These are two crayfish here that are actually fighting. We do a lot of social dynamics out there because crayfish, when they fight and interact, they exchange chemicals back and forth. So if there's chemicals, there's got to be flow. This right here is actually a particularly nasty chemical. This is 2,4-D. This is used in a lot of things like Roundup, and when we put it on our lawns, it flows into the water. So we are interested in what this does to these animals and other types of animals as it gets into a stream system. Lots of organics, and finally we're starting to get into these types of things as medical waste gets into aquatic systems. And they have really interesting impacts on aquatic organisms. We can do these kind of controlled studies out of the stream lab, and we've used all these chemicals at some point over the last 20 years. Now, I can't cover all the work we've done, but I can show you some of the stuff. Now, what's interesting, if you study chemical signals, the hardest thing about studying chemical signals is measuring them at the scale of the response of the system, right? We can measure light pretty easily, we can measure sound pretty easily. But these systems, whether it's an algal cell or an animal or a fish or a macroinvertebrate, respond to chemical signals on the order of seconds to milliseconds. I've been lucky enough to be in the right place at the right time that I collaborated with a guy who took an HPLC, turned it inside out so that we can take that detector, put it out in the field, and we can start to measure chemical signals at the spatial and temporal scales that animals respond to them, and that's what you see up here. This is actually Dave Edwards' work, and this is an old student of mine. This is the Maple River, this is Carp Creek, and this is what the system looks like. This big thing is an ADV, and it allows us to measure the three-dimensional dynamics of flow. What you don't see at the end of this is a carbon fiber about the size of a human hair. And what we do is we hook this carbon fiber up to a computer system, put it in the middle of the river, and we charge that fiber with a voltage. And then when chemicals come by, organic chemicals come by, they hit that voltage and they get oxidized, and we measure that as a current. So what happens is if you happen to be an organism sitting here and something gets dumped in here, one of those medical wastes or nitrogen or phosphorus or 2,4-D or something like this is what you see downstream. We have the ability now in stream systems like this or at the stream lab, when we ask an animal, how does this chemical impact you, we have the ability to measure that chemical. It's pretty obvious. If you're going to change a system, you want to measure how you change that system. So now we have that ability. And for the last 10 years or so, we've been going out and not only looking at the impact of chemicals on the system, we've been measuring that impact on that system. And what's interesting about it, here's a movie just to show you what this is like. Maybe. Maybe not. All right, just imagine this is playing. All right, what's interesting is if a chemical comes in, constantly being emitted, like a fish has died or farmers put fertilizer on, what you see are these little wisps of chemicals. Okay? So if we look at what an animal is actually responding to, what we often measure, right, is this average, and that's what this red line is. But what the system is seeing is not the average. So when we go out in our summer courses, we take a water bottle and say, oh, let's look at the nitrogen in that water bottle. That's not what the system responds to. The system is responding to this stuff above the average. And that's what we're doing in this system. Okay. This is some of Troy's work, if he remembers this. We can ask questions in these flumes about the interaction between two types of odors. So he's looking at predator and predator and food odors and how they interact with crayfish. We can look at gutter systems. So these are stream crossings. We can put these out at the stream lab. We can put Y mazes out at the stream lab. You can use elevated temperature experiments out at the stream lab. Habitat choice, and finally a whole bunch of soil behavior. So we have a lot of different dynamics of things we can do. And just really super quick. This is a recent study. This is a lake. This is a river. Crayfish live in the lake, live in the river. Fundamentally, the difference between lakes and rivers are flow, right? So we ask a simple question. Put animals in a flowing system. We ask the crayfish, can you find food? And we turn the flow off. And we take animals that have lived their entire life in lakes, animals that live their entire life in rivers, and ask them an identical question. What's intriguing is that, really quickly, okay, river animals that are over here don't change their behavior in a river or a lake, but lake animals do. So we're beginning to ask evolutionary questions about how the habitat is structuring the behavioral response to chemicals. All right, so that is 20 years of work in 10 minutes. So I'll take some questions if you want.
We do have time for questions.
Okay.
Comments or questions, Paul?
How concerned are you about the possibilities of something being dumped in the river there at Douglas Lake Road? You think that's salt, and they salt the road and stuff like that?
We, not so much the stream lab, but the Limno course 4 years ago worked with Paradise Lake, and they've got a major problem. It's 31 runs right by Paradise Lake, and they've got a major problem with salt influx. So the salt influx from that road coming into the lake changes the chemistry of that lake and has an impact. So I'm not so sure about stream crossings because, you know, you've got about 5 meters or so, but a large stretch of the highway really impacts those dynamics.
Thanks. I think we have to move on to stay on time. But I want to just, as you talked this afternoon in the aquatic breakout sections, I'd like you to think about what improvements we might make. We have opportunities to write proposals at NSF and other sources. So think about that. Okay, our next speaker is Paul Drevnick, who was our most recently hired research scientist at the station. Hired on Pi Day.
Where do I put this?
Somewhere.
I wanted to hire him on March 15th, but he insisted on Pi Day.
Yeah. No, your stuff. There you go.
So I have 1 year's worth of work. I can for sure do that in 10 minutes. Although I thought I had 15, so we'll see how that goes. So we're starting watershed research at the biostation to couple terrestrial and aquatic systems. I say where is the water because the water is not evident in our watershed. So I'll start with acknowledgments. Funding is from the McIntyre Stennis program of the U.S. Department of Agriculture. Co-PIs are Luke Nave and Bronco Kirkes. Bronco is an engineering professor, and he'll actually get all of our measurements continuously online when we really start them in mass this summer. It's great working with Luke and Bronco and Jim Lemoyne and Jason Talent and Bob Vandekoppel, students. It's really a great collaborative environment that we have at the biostation. Bob isn't necessarily very supportive of where we've chosen to do the work.
Don't forget he's watching.
Oh, I think he'd agree with me. And the reason is we're studying a stream or a stream in a watershed, and the stream isn't very impressive. But I think it really turns out that in the studies that we're doing here at the biostation, the surface water stream isn't all that important to the movement of water. It's what's in the ground, below ground. I thought since I've been here for about a year, I would go over my expectations of my job. And that's a research program that focuses on environmental problems in the Great Lakes. And one of those was using watershed approaches on energy or materials exchange between terrestrial and aquatic systems. My research program, I've made goals of increasing knowledge of the station's aquatic resources. One of that will be writing a review of what's been done, and then going forth from that. I'd say at this point, I would also like to be a resource for people about the aquatic resources, although at this point, it's the people here that are more resources to me, that are more knowledgeable about the study sites. The other goal is to address problems of water quality in the Great Lakes Basin. A very smart man told me that the large lens of groundwater under in the thick, sandy, gravelly glacial deposits overlying sandstone and limestone bedrock interacts with all manner of surface water, vegetation, and various life forms in the tip of the mitt region at the confluence of the three largest of the Great Lakes. You did say that.
Okay. Thank you.
It's unlike any other aquatic system that I have worked in. I've worked in aquatic systems, or at least the watersheds where it's shallow glacial deposits with impermeable bedrock underneath. It's a challenge, and we really need to understand the groundwater in these systems in order to understand how these systems function, especially for the lakes in the region. So this is a figure that's 20 years old, no, actually, from an SNRE student. And the take-home message here is that in the Sheboygan River watershed, where the biostation is, has the greatest relative groundwater loading rates anywhere in the lower peninsula of Michigan. What this is it's soils with high porosity, so it's sandy outwash soils that the water just rushes through, and we have elevational gradients that move that water. Bob Vandekoppel recently shared with me his hydrologic budget for Douglas Lake, which he just punched these numbers in from last winter, an even colder winter than this one. And what he actually estimates is that the groundwater inputs, at least for last winter, were on the order of 168 to 241 million cubic feet per day going into Douglas Lake. That's a tremendous amount, and it's the biggest term in the hydrologic budget. What that says to me is that the groundwater is extremely important, and we need to understand the groundwater for management decisions that involve our surface waters. In the Sheboygan River watershed, we have groundwater-dominated headwater streams that are getting a lot of that groundwater, and it supports cold water fisheries. So all of the streams highlighted there in blue are cold water fisheries in the Sheboygan River watershed. We also have lakes, so the big lakes in the watershed. I suppose I could use this on which. Yeah, wrong one. The lakes here are, so Douglas Lake is here. They're receiving more groundwater seepage. So that's not non-channelized flow. It's a more important renewable water source than the streams are to the lakes. So like I said, management decisions regarding land development and uses of groundwater must take into account the importance of groundwater for maintaining high water quality in streams and lakes. Also, in the Sheboygan River watershed, we have a mercury problem. So Crooked, Pickerel, Burt, and Mullet Lake are all listed as federally impaired for exceeding the U.S. EPA water quality criterion for methylmercury in fish tissue for the protection of human health. I'm convinced that Douglas Lake is not on that list because it doesn't have a DNR boat ramp. The mercury levels that I've measured in these fish are higher than in the other lakes. So our initial watershed study is focusing on understanding this mercury problem. I can maybe best explain the mercury problem with this research that I've done at Isle Royal and a literature review actually. So another location in northern Michigan. The mercury problem is really mercury coming from the atmosphere, from say coal-fired power plants being deposited in our ecosystems. At Isle Royal, which is a not very disturbed environment, if we take mercury in sediment cores and we use that with a simple model to predict atmospheric mercury deposition, what we have is here, more or less an increasing trend throughout. On the other hand, if we take mercury in moose teeth from Isle Royal collected in this period, what we see is a decline since about the 1980s. If we actually look at measured mercury wet deposition, we see something in the middle and we see more of a declining trend. What this suggests to me is that the sediments are overestimating the atmospheric deposition of mercury. And the reason is we're having mercury that's in the watershed go into the lakes. It's either a legacy effect from previously deposited mercury, or it's from ecosystem changes that's making mercury move faster from being deposited into our lakes and accumulating in our fish and causing the mercury problem. Our main hypothesis for the watershed study is to test the null hypothesis that watershed retention rates of mercury have not changed with time. This is another statistical model where we're taking, for different lakes, watershed area to lake area ratios and regressing it against a pre-industrial mercury accumulation. And we can do this for boreal lakes in Michigan, pre-settlement. So Denny Albert did a lot of this work to define where these are. If we look at retention, what we take is the slope divided by the intercept, and for boreal watersheds, it's about an 80% retention of atmospheric mercury. If we look at these others, which would be more hardwood or mixed forests, it would be a 96% retention of mercury, atmospherically deposited mercury in the forest soils. We're not getting that high of retention anymore, I think, and we'll be testing that hypothesis. So, for our watershed study, we'll be monitoring inputs and outputs of mercury and carbon. The mercury in the watershed is moving with carbon. We've chosen this watershed here. The watershed boundary is delineated here. It's a first-order watershed that's entirely on biostation property that's draining into Burt Lake, one of our federally impaired water bodies. There's a close-up. So we must understand the hydrology of the system before we can look at mercury and carbon cycling. So we define that topographic boundary on the previous map, but we've also got to define the phreatic boundary, which is what we don't see underneath. So glacial deposits or bedrock that could be having water come from a larger area. And that's a problem in Michigan in defining a watershed. So we'll also be monitoring precipitation and evapotranspiration. Those are easy to do at the biostation because they're already done for us. And then stream discharge. But because we are in violation of an assumption of the small watershed model, the assumption here would be shallow impermeable bedrock. We also have to monitor this non-channelized flow to the lake that's not in the stream, and then loss below. There's actually one slide of data, but we can skip that. So what we've been working on is actually characterizing ecosystem components, so soil, above and below ground biomass, by going back to these landscape ecosystem plots. So our watershed right here is right here. We're on outwash, going up a moraine. Eventually we'll be doing watershed scale studies to understand inputs and outputs and retention of mercury. We'll be doing plot scale studies to understand processes that affect the retention. We'll be adding other watersheds, I hope, with more funding and more time. Our goal is to understand forest management practice that maximizes mercury retention in the soils. And there are opportunities for collaboration here, especially understanding the hydrology of the system. I'm not a hydrologist. And with this watershed study, we want to branch out, not just focus on mercury and carbon, but other things. Thank you.
Thanks, Paul. This is the time for a question or comment.
Danny.
Just a quick comment on I was talking about Great Lakes, and one of the things that's interesting in the Great Lakes shorelines in these last few years of low water is all of a sudden you realize that the groundwater is playing a major role when the Great Lakes water levels drop.
Yeah.
And so it's a question of that's another area close to the station that you could be working in this groundwater.
Absolutely. So in the Great Lakes hydrologic budgets, groundwater seepage or deep groundwater inputs are not included in those budgets at all. And we know for sure for Lake Huron, where there are those huge groundwater seeps that are supporting unique ecosystems. And so it's something I definitely want to work on.
Thanks, Paul. That was great. I think this is a new dimension. And Paul and I have talked about this in several of you, but in the breakout session on aquatics, the Geosciences Director at the National Science Foundation has a program called the Critical Zone Observatory. And basically, the focus is on that groundwater. But if you read the program description, it's focused on more than just the groundwater. It's basically the critical zone is viewed as everything from the top of the canopy to bedrock. And I think this line of research, I'm happy to see Luke and Paul have started, but this could form, if you'll excuse the expression, the bedrock for a critical zone observatory proposal the next time there's an RFP, which if things are going along the normal time schedule, will probably be 3 or 4 years from now. So I think this is an opportunity for big work. And Luke has a loud voice, so he doesn't want to use this. And if you can't hear him, just... Okay, Luke, thanks.
Thank you. My title is new in quotes because, as many of you know, there's nothing new under the sun. And this is basically part sales job. Paul took care of part of that. We need people doing hydrology in particular on this study. And also an admission that I'm very much interested in moving in the geo direction in this study, becoming more of a geoecologist than the bioecologist that I've largely been. The background for why we're doing this is the motivation. Knute set that for us, hiring Paul, soon to come, atmospheric research scientist. We're all ready to work together and write grants and start studies that are integrative and interdisciplinary, such as the one that we're conducting in Honeysuckle Creek watershed. The foundations we're building upon, the many things done by other people that we depend on, is the landscape ecosystem framework that Bert Barnes and Led and many other people have participated in, the carbon cycle research program at UMBS is very important to our understanding of this watershed that we're studying, atmospheric deposition, which has been ongoing for a very long time. And also, I didn't appreciate the many uses of LiDAR data. The LiDAR data set that we have for UMBS is very valuable not only for canopy structure in the sense that it's been used largely so far, but for digital elevation models and understanding the surface of the Earth that we're interested in. Sales pitch, we have opportunities here, and I'd like to extend them to you and people that you know to study aspects of this watershed, the Honeysuckle Creek watershed. The primary research questions that those of us who've started this are interested in are largely pertaining to carbon and mercury, distribution, cycling, movement between the ecosystem types that comprise the watershed, and the processes that operate within soils, within ecosystems that transfer carbon and mercury between ecosystems, that transform it between different chemical forms. We've chosen to do this in the Honeysuckle Creek watershed. Here in southern Michigan, honeysuckle has a bad connotation. We think of Tartarica, other non-native invasives, but at the biological station there are native honeysuckles that are great indicators of the calcareous till, the parent material in large portions of our watershed, and which indicate things about the flow of water, the chemistry of the soil. This watershed is 80 hectares in size. There are 13 landscape ecosystem types in it, as classified, summarized by Doug Pearsall in his dissertation. The plant cover types with which many of us are more familiar, anything from xeric to mesic aspen, northern hardwoods. A large area in the lower watershed is mixed conifer hardwood swamp. Top of the watershed is 275 meters at the Pelston Hill, drains down to 181 meters into Burt Lake. The landforms, the physiography of this set of landscape ecosystems covers almost all of the landforms present at the biological station along the course of the stream here. We look to the west, a visualization here of the topographic wetness index overlaid on a 3D DEM, showing the high point here on top of the Pelston Hill and flowing down. Here's West Burt Lake Road. Here is a large area of mucky cedar swamp. Parent materials are many, in keeping with the number of landforms, everything from the calcareous till that is the core of the Pelston Hill, the gravelly and sandy outwash deposits that those of us who have studied the Ameriflux and facet footprints are more familiar with. There's also aeolian deposits, old dunes, lake bottom sands, and also the organic deposits. It pains me to not spend 10 minutes on this slide because this is what I'm talking about with geoecology. When you look at the nature of the surface of the Earth, where we do our research at the biological station, you can tell so much that I, for one, didn't appreciate for a long time. This is based on the LiDAR data that Brady Hardiman obtained for basically the Ameriflux footprint and surrounding area, only this is the 3D digital elevation model. You can see things, the gorge jumps right out at you. The big Pelston Hill here, Riggsville Road basically follows this line. We've got Bryant right here, the meltwater channel that drained the ice contact area up by Ameriflux plot D9, if you know that area well. Pelston Plain over here. Anyway, point being, we are approaching this work from the ground up in terms of defining our watershed down here, which is contained by some old dunes on Algonquin lake bed. We're looking at the geographic basis for this and approaching it in the landscape ecosystems framework. You've seen this picture already. It's Paul's. This is what our stream looks like, at least in the portions of its reach where it's well behaved. It'll hit a log and just disappear under the ground at times. I'll show you some data. This is Science Light as well, kind of a diet version. There are some numbers in here that explain why Bob recommended against doing this study here. What we've done so far last fall is to revisit 5 of the landscape ecosystem soil pits, or the plots where the pits were dug, redig those pits and take new profile descriptions. We deployed some sensors that Paul talked about. Right now, we've got UROP students working on analyzing the soil samples. We're mapping the soil moisture and the thickness of the muck deposits in the lower part of the watershed. And in 2015, we're going to do a high-intensity soil survey and surface deposit measurements, and also more intensive soil moisture measurements. Results light. Yep, here it is. A little bit of science to keep you interested. What we have here is 5 landscape ecosystems plots with old. That's basically, the convention here is year, 1988, plot number 6, 1991, plot 51. So we have plots visited about 15 to 25 years ago. Measurements were taken of the O horizon, A horizon often didn't exist that time, and then a measurement that we took in September. The point I'd like to show here is that O horizons are being converted into A horizons over the time period. Some of this could be differences in methods. There may have been small amounts of A horizon material recorded by the Pearsall-led crews or all the people that worked on the landscape ecosystems, but even if you account for that and say, let's talk about the O and the A horizon as representing the organic rich topsoil portion of the soil, we've lost 1 to 3 centimeters of that material, which accounts for about 5 to 10% of 1 meter soil carbon storage over that time period. That's probably a big deal in ways just beyond carbon and mercury cycling. Point being, that was upland stuff. This is where I've cut my teeth. This is what I'm comfortable with. But what we really need to understand is places that look like this, places that look like this. Perhaps you can see why I'm not particularly eager to go into that sort of ecosystem, but this is where the interesting stuff is happening for mercury transformation, for carbon accumulation versus loss. So a couple of results light about the wetland ecosystems. We compare the peat measurements, actually the muck measurements, importantly, we took in October with UROPS. For type 118, this is the mid-moraine channel hardwood swamp. For those plots within that ecosystem type in the honeysuckle watershed, we find a conversion of what was once sapric muck, well-decomposed organic matter, to more of a mineral muck mixture of mucky sand, deeper in thickness, representing some incorporation of what once accumulated as surface organic matter into a more mixed mineral profile. Also the water table, and here's a point that Bob has made again and again. The last 2 years have been incredibly wet, and not only have we gotten like 40 inches of rain, this is huge. It's the timing of it. It has happened in large part in the fall after transpiration has turned off, so that water recharges aquifers, it runs off. Point is, the water table depths that we measure last fall are well outside of the range of what Pearsall, Greg Zog, other people in the 90s and 80s recorded. They refer to water table depths below 40 centimeters in this ecosystem type. We found it either at the surface or as deep as 25 centimeters when we surveyed in September and October. The conifer swamp, basically the same story. We've got a conversion of more pure, undecomposed peaty materials into a more decomposed sapric state. The thickness hasn't really changed. Again, the water table is high. The organic deposits, I think these represent historic accumulation. We're in a period in the last 20 or 30 years where what was built up over decades or centuries is being mineralized and moved. This is important in terms of what it's doing to transferring carbon and mercury from upland soils where they were accumulating into highly reducing conditions of wetlands where mercury methylation occurs, where carbon transformations occur. We've got a whole bunch of hypotheses we've thought about that basically all relate to carbon and mercury. Not all of them are testable. We do have plans for how we will test some of these. I've showed you some of that. Revisiting the soil pits done by the landscape ecosystems work is a big part of that. We've got this wealth of past data. We can go back to the same places and compare. We can also use gradients within the watershed, wet to dry areas, to understand how increases in the water table or decreases in the water table affect things like mercury methylation or carbon accumulation versus loss in organic soils. We've got plans to collect a lot of data on basically biogeochemical stuff, but a lot of this will emphasize, from my point of view, the soils, the nitrogen and carbon transformations. We can't do any of this without other people that are as interested as we are in understanding this little patch of the earth. So it's an open invitation to bring what you know. If you're interested in this place, work with us. And of course, rub for some of that sweet cash money at every step of the way to fund this fantastic work. When I've said us, I mean a lot of people. There's a number of people, collaborators, investigators, what I came up with, people who have helped out by generating data and given some time to this so far. We've got great UROPs and, of course, people who have imparted information that they have about the place we're studying that helps to understand it. And the biostation, that biostation is good. All right, that's what I got.
Thanks, Luke.
Thank you.
We do have time for a comment or two or a question.
Comments, please. Yeah, Danny. Uh-oh.
Yeah, do you have any plans for manipulations on the watershed?
Plans? No. We've thought about it, but no plans.
Concrete plans, but I think eventually it will become part of the study design.
Oh, the reason I ask is because in some of my coastal studies, I've been forced to study in an area that has been diked versus not diked.
Mm-hmm.
And when you talk about those mineralizations, 50 meters apart, you can find soils that have, in those last 15 years of dry conditions, have completely, the organics have completely mineralized, whereas in that dike, where they've been keeping it high, you've still got 40 centimeters to 50 centimeters organic. So you might be able to do some small manipulations to address that.
Thank you. That's good.
Manipulate.
Do I get to ask? Okay.
Thank you. Plenty of information and ideas for carrying through this afternoon. I'd like to introduce Bob Fahey. Bob. Oh, there's some empty seats up here.
In the second row, it's
Yeah. So if you're, and how's the volume in the back? Good. It's okay when Luke was talking, so the room works pretty well. I want to make sure that people who are online can hear it, so I'd like to encourage people to use the mic when the mic is working. We think we have it working now. So Bob Fahey has been working and teaching at the Biological Station for 3 years, and 4? Time goes fast when you get his old time. And he's from the Morton Arboretum in Chicago. So Bob.
All right, thanks. I'm. A lot of this has been like, oh, this is what we're going to do. We're excited. We're going. This is stuff that's a little bit more farther along. We're talking about the Facet project, which Chris has been working on for decades. It feels like so. And Peter. But so I'm going to talk a little bit about community responses to the Facet disturbance. So most of you are probably pretty familiar with the Facet project. But just to reiterate what the disturbance actually was, so all the aspen and paper birch, the early successional canopy trees were removed by girdling in 2008, and that resulted in gradual canopy loss over multiple years, which has resulted in, well, it was about 39% of the basal area, and it took a couple years for it to reach peak LAI loss. So in 2010 was the peak LAI loss in the system. And this was always sort of designed, I think, as a successional transition, but it also works really well as a model for what happens when you have canopy loss due to introduced pests or pathogens, or a rapid canopy senescence that might be precipitated by something like an environmental, like a drought or something that might lead to really rapid canopy senescence. And the goal of the work that I'm going to talk, the goal of the overall project is to understand the ecosystem response to that moderate severity disturbance, that successional transition. And as Chris will probably talk about later, I think they've seen pretty high resistance of the system to that disturbance in terms of ecosystem functioning. Up to about 60% basal area loss. This is from Ellen's work. And Chris will probably, I think, go into more detail on that. But at really high basal area losses, you don't see that same resistance. So the work that's been going on has been, mostly done by ecosystem ecologists like Chris and Peter, who see the forest as just a big leaf that produces carbon to go up to their tower, right? No, they're more nuanced than that. They are. But the community response, so how the actual community composition and structure of the ecosystem responds is going to be really important too. And I know they recognize that. But one of the main reasons is that a lot of the landscape of northeastern North America is going to undergo this similar sort of transition. Maybe not so quickly, in some places maybe even more quickly, in the next few decades. So this is the age of the biostation forest. This is the average age of northeast forests. So they're going to reach this successional transition where they go from an early successional canopy to a more mid to late successional canopy soon. The yellow up here is aspen and birch forests. A lot of those are those early successional species, successional systems that are soon going to transition to that later successional composition. And we also have things like emerald ash borer here that are transitioning ecosystems quickly from one species composition to another and creating these moderate severity disturbances. And what we've been seeing across the eastern U.S. recently is that most ecosystems are becoming maple dominated because of the loss of fire from our landscapes and this process of mesification, which I'm not going to go into. But we see these carpets of maple everywhere, and you don't see a lot of the shade intolerant and mid-tolerant species that used to be a pretty strong component of the overall community. And this moderate severity disturbances from a bunch of research seem to be really important for maintaining some of these mid-tolerant, mid-successional to late successional species in the landscape. So things like white pines and oaks are maintained by these moderate severity disturbances. Community response is also going to be important from an ecosystem perspective. So the sub-canopy contribution to NPP seems to be pretty important in the resistance of the system to the disturbance. And these are graphs from Ellen that show the fraction of senescent basal area and the sub-canopy contribution to ANPP. So the sub-canopy contribution to productivity increases as you get higher severity disturbance. And that's related to species-specific responses, physiological responses, and morphological responses. There's Ellen measuring these things. And so we know that sub-canopy productivity is going to be related to disturbance severity, but that's not a very strong relationship. You would expect probably a much stronger relationship. One of the issues is that there's a couple plots here that were past this sort of disturbance severity threshold that for some reason the canopy didn't respond because the canopy is mostly gone, but the sub-canopy also didn't respond and isn't very productive. And we don't really know why, and that's something I think we need to dig into a little bit more over the next couple of years. So these two plots here, that the canopy is mostly gone, but the sub-canopy didn't respond, and it's probably got something to do with the structure of the system. But even when you take those outliers, if we can call them outliers, out, it's still not that strong of a relationship. So sub-canopy productivity is being driven by something other than just the disturbance severity. And so we wanted to know, well, how strongly is that sub-canopy response to the facet disturbance related to composition and structure, so the community composition? And then how is the facet disturbance then affecting species composition and community structure in this system? And then I'm also looking at some sort of spatial patterns in growth and competitive interactions within the system and how they're affected by different disturbance severities. So this is building on a bunch of REU projects and some other work that we've done. So Liz Stockton in 2012 remeasured all the saplings in facet. Aubrey De La Cruz in 2013 measured all the saplings in subplots within the facet plots and took increment hammer cores from all of these subcanopy trees to look at growth patterns over time. And then there was a bunch of physiological monitoring done in 2014 and also in 2013 by Ellen and Rick Beckel, another RU student. So in terms of this first question, so how strongly is subcanopy response related to composition and structure? This was designed to break your brain. So hopefully it worked. No, so this is an ordination of species composition in the subcanopy in the facet, across the entire facet system. And basically, there's sugar maple dominated plots, there's red oak dominated plots, pine dominated plots, beech dominated plots. And this is that same ordination with understory productivity, subcanopy productivity overlaid. And what you see is that oak and sugar maple dominated plots have much higher subcanopy productivity. And this axis here, this first axis from the ordination is a really strong predictor of subcanopy productivity. So there's pretty good evidence that it's not just a disturbance severity response. The community is really mediating this response. But disturbance severity does matter. So if we look at the growth rate of large saplings, so the bigger trees that we took cores out of, as you look at how much basal area was removed versus basal area increment for those trees, there is a pretty strong relationship in both total basal area response and then average per tree response related to disturbance severity. So when you have higher disturbance severity, you get higher productivity. But at the same time, composition is also really important. So this is that same ordination axis. So these two things are kind of working together, but then the composition is also responding to the disturbance. And I'll show you a little bit of the results on that. So this is just a picture of these, where the trees are, the subcanopy, are the subplots within the larger facet plots, these little clover leaves. When you look at plot level sapling biomass, so we're interested in how the community might change in the future related to this disturbance. This is 2000, the blue is the 2006 biomass, red is 2010 biomass, and green is 2012 biomass of subcanopy trees. So I can't remember the size classes, but what we see is that red oak was a pretty small component starting off in 2006, but has gone like gangbusters since then. We see a lot of productivity from red oak, and some of these the maples aren't quite as responsive. Beech, there's a lot of it, but it's not really responding to the disturbance, partly because a lot of the beech is in places that didn't have much canopy loss. This is the growth of those large saplings that we took cores out of. These open circles are the pre-peak basal area increment per unit of basal area. So how fast they're growing relative to how big they are. And so pre-peak, we already had pretty fast growing red oaks. The red maples were also growing pretty fast. The ironwoods were growing pretty fast, but there's very few of those. So it's not really that important of a component. And then this is the response. So this is the post-peak basal area growth. And you see red oak, again, very responsive to the disturbance. And this is just basal area increment over time for the different species. So here's red oak, here's red maple, and then everything else is pretty minor component. And you can see that not only is red oak growing the most, but it's responding most quickly. And this is at a plot scale. So a few of these plots seem to be becoming red oak dominated in the subcanopy. There's a few other plots where red maple is dominating these dark red bars, and it doesn't seem to be all that related to disturbance severity. So actually the red maple dominated plots had the highest disturbance severity, which doesn't really fit what we would think because red maple is a little less of an intolerant species. But we do think that it does seem to show that red oak is going to become a more important part of the system, which is very different from everywhere else in the entire northeastern U.S., that we're getting this strong red oak response, which is almost certainly something related to the environment here that allows red oak to be competitive with the maples. But the competitive part of things is the next thing we're looking at. So we want to look at how that wood production that was going to the aspens and birches has been redistributed to the other trees in the system spatially, and relate that to disturbance severity and the patchiness of the disturbance, and also to the site productivity. So that's the next step of what we're doing, to do some neighborhood analysis on growth and productivity for canopy and subcanopy trees. But I'm not going to talk about that because I haven't done it yet. But I'll be working on it this summer, and hopefully with some students. So with that, I'll take questions.
We have time for a comment or question or two.
Have you thought about doing some of the growth increment analyses in the treatment replicate stands, some of which are in different ecosystem types, and might be reacting differently than the dry outwalk where most of the treatment footprint is located?
It's a good point. Something we should do. So which stands are you talking about?
These are the 2-hectare girdled areas in the Ameriflux footprint that are in types 59 and 116. They're different than the facet footprint, the main area.
Yeah, I mean, it would be good to get, so right now the subplots were the ones that Ellen worked in, and they were sort of very much constrained in terms of the environment. So it'd be good to expand this even within facet to some of the plots that are maybe farther out and are more, maple dominated or beech dominated. So yeah, so far, yeah, it'd be good to do this in those other plots eventually. It's a lot of work though. We got lots of RU students though.
Question, Bob, for you and Chris, I should know this, but the subcanopy, is it really focusing on the shrub and sapling layer, or are you also or is Ellen also looking at the ground layer vegetation?
No, this is focused on trees.
Okay.
Yeah.
Okay. So
it'd be good to look at some of the ground layer dynamics too.
Yeah.
There is production going on there.
Yeah. I think a couple of us in our lab, Jacqueline Poplar might be interested in talking with you and Chris about collaborating on the ground layer vegetation. Okay, thank you.
Thanks.
Next speaker is, I believe, Brian Scholtens from the College of Charleston.
Yeah, thanks for having all those nice cold weather for us.
It disinfects you.
It does. So laser and spotting things. Okay, very good. Thank you. So I should start up by doing this because you'll see a bunch of hand waving up here too. So, sorry, go ahead. And this is a summary of. So we were talking about long-term data sets. So I probably get to beat everybody here because some of what I'm talking about is at least 70 years. Do I need this?
I think if you can, because people are watching on the web.
Oh, okay.
Better for them.
We'll try it here. Okay. And what I've done here is tried to give a very broad summary of insect knowledge in the station region, what we know right now, and where I think would be interesting to try to go, although it's going to be difficult to get there. And this is other stuff that we know pretty well from the station region. These numbers, I'm sure, are not exactly right. These are from lists that Bob Vandekoppel maintains, but at least they're close, right? So fishes, amphibians, reptiles, birds, mammals, mosses, even mosses, right? Well, who did that? Vascular plants, and algae, right? So Rex and Pat have done a great job with algal surveys, and we have pretty darn good idea of the algal species that occur in various habitats, aquatic habitats at the station. So Bob often gives me this kind of a question in some form, because he's often approached by people asking him, does this species occur up there? Or what do we know about the species in this kind of place? And he usually passes that question on to me, and I have to tell him with a lot of hand waving, well, yeah, it's probably here, and it might be. So basically, we don't know, and this is one of our big problems. But we do know. We have a great survey of odonates, of dragonflies and damselflies. We probably pretty much know every species that's in the station area for dragonflies and damselflies, because we've had some really good researchers over the years work on those. We know butterflies. This is primarily Ed Voss and myself. That's going back about 70 years. I can't give you a number for really anything else in the insects, where we actually know the species that occur in the station area. So this would be really valuable for a bunch of people. Anybody who's doing any of these kinds of studies, succession studies, if you're interested in insects that live in the soil, and I know they're just getting started thinking about this with a burn. So what do we know about insects in the soil? What do we know about ground beetles? What do we know about ants? We basically don't. We have very little information about it. Pollination work, what Claudia is going to talk about later on, knowing just a little bit about what the potential pollinators are in these systems would be really valuable when you start and you're trying to figure out pollination systems. Feeding studies, I've been involved in several of these, looking at plover diets and bat diets and pitcher plant diets even. And just knowing what are they actually eating, what are the possibilities for them to be eating, and we don't know that much. Climate change studies is another one. We have a few tiny hints in insects with climate change studies. This is an insect that, unless you spent any time out west, none of you have ever seen. I've never seen this insect, giant lacewing. But if you go into the UMBS collection, or if you go into the museum's collection just a few blocks away from here, you'll see that this was collected regularly at the station through the 1950s. And then it disappeared. This is an insect this big. We have no idea why this disappeared. In fact, people did not notice that it disappeared until the 1970s, because nobody looks at it. So it disappeared not just in Michigan, but throughout the eastern United States. It still occurs in the western United States, but we have no idea why it disappeared. Was it something to do with changing habitats? We don't know. We have no clue. We do know, at least a little bit, based on work that Ed Voss has done in the area, that we have had some movement of butterflies, one of those groups that we know something about. These three skippers have all invaded the area from farther south. So when Ed Voss was doing all of his butterfly work, and we have excellent field notes from his butterfly work, when he would go to wetlands, he would record everything he saw, everything he collected, and these three species were not among them, all the way through the 40s, 50s, 60s. In the 70s, we first picked up the first one on the left, the broadwing skipper. In the late 80s, we picked up the Dione skipper. And 2 years ago, we picked up the black dash. And if you look at all the old maps, the closest these had been previously recorded were about 4 counties farther south. So nobody is looking at butterflies between there and the station. And so they've obviously been creeping up, and they now all occur as far north as the Straits. So at least a few species we know have been moving. But that's about as best we can say for anything for climate change for insects in the station area. So what I wanted to do was find out what really we don't know. So at the end of my insect class in the summer, I always tell the students, one of my major goals is for them to know how much they don't know. And I decided I needed to do a little self-reflection and find out how much I don't know. And that's what I tried to do here. So over a period of years, I have surveyed major collections. Now, not completely by any means. I haven't gone completely through the Michigan collection here for station records, but for at least select groups. and examined literature records from the Voss papers and a whole bunch of other papers for finding out species that are there. And I've started using my class each summer to focus on what I considered some undersampled groups to try to increase our knowledge of those groups in the station area. So where'd we end up? So here's my hand waving. There's a column there for North American species numbers, right, for insects. So North America is about 100,000 species of insects. There's a column there for what I've found recorded from the UMBS region. I've recorded currently about 3,850 species of insects. Not all these are identified to species, but we know they're different because they're in a family that, you know, it hasn't been recorded. So, and you notice that that means we have recorded about 4% of the North American fauna in the station region. Now, who knows? That may be great. That may be normal, right? But then I started thinking, well, so how do I actually estimate what we think should be there? And what I did was think, okay, what groups do we know? And the groups that we know are those two, which are the odonates and the Lepidoptera, primarily because of Ed Voss and myself sampling these over many, many, many years. There's the 70-year part of this. And we know the odonates are about 18% of the North American fauna, 18 to 19% that we know. And the Lepidoptera are, this is butterflies and moths, about 13% of the North American fauna. So I figured, let's split the difference, because I think odonates are actually overrepresented a bit in the northern regions. And I know that we're not quite there with the Lepidoptera yet. So I figured, let's use 15% as what we would estimate a typical group would have in the North American fauna in northern Michigan if it's completely sampled. Okay? And you notice that none of the others are even close to that in terms of how much sampling we actually have. So if we do that, here's what we get for our estimate. We would estimate there should be 14,000 species of insects in the station area. And we currently have a list of 3,800. And 10,000 of those, 10,500 of those are the ones we haven't found and recorded yet. So we have a long ways to go to try to figure out what's going on with the insects of the station region. And you notice those are my two numbers that I use to split the difference, right? So that's assuming that odonates are oversampled for the area compared to North America, and the Lepidoptera are just slightly under. So we're sitting at about 84 to 90%, depending on what numbers I use, for how many moths and butterflies occur in the station area. It's pretty well sampled. So where do we need to go? Well, one of the things I find out is that the current levels of knowledge in our collections are really miserable. The taxonomic, they're in a mess. So it takes a lot of sleuthing to try to figure out what's going on when I go into the collections and try to get the data. You wouldn't expect that, but it's true. Lots and lots of old names that don't apply anymore that I have to try to figure out and track down what's going on, what it actually means now. Lots of things that are misidentified. It takes quite a bit of work. Databasing would be really valuable. And we've piloted some stuff with the class with Jason that developed the database this last year to be able to start trying to enter these data into a larger scale database for the station. We're going to continue trying to do that this summer. I'm going to try to add in with him about the 6,500 Lepidoptera records that we've got for the station area. And we're just starting to see hints of some patterns in the stuff that the students have done, concentrating on Wilderness State Park. Because we're getting, just like with the plants, obvious southern and western affiliations in the insects of the dunes at Wilderness State Park, the same way we would do with the plants. This is just a screenshot of the data database that Jason's been working on, where we can plot our data, and those are some insect points from my students last summer right at the station area. But it stretches over the whole region, and we can plot any of those things. Other ways, we also need to try to get some groups sampled better that might help us out. Even things that we think might be well sampled that have had experts there aren't necessarily so great. So Hungerford, and I wish I had a better image of his book here, but Hungerford was at the station for 30 years. His group was Hemiptera. Now, he focused on aquatic Hemiptera, and I didn't break that out. But Hemiptera only have about 20% of the samples we expect in terms of species that we've recorded. So we obviously need some efforts, even in places where we think we've had big experts in the area. And lastly, we need to go there and evaluate the needs of all of you. What kinds of things would be best for you? One thing that pops out at me immediately is we need better samples of aquatic insects. And we have large aquatic groups of researchers. And face it, when you guys go do your aquatic insect work, you try to do samples, what, family IDs, genus IDs, maybe? We don't know the species that are up there. And just to show you that, there's the three major aquatic groups, right? And so they are sampled at about 21%, 6%, and 11% total sampled based on these estimates. So we really need to, and those are major groups used in aquatic monitoring. So if you are somebody who monitors aquatic ecosystems, everybody knows what an EPT index is. That stands for ephemera, plecoptera, trichoptera. That's those three groups. That's what people use as their monitoring index. And that's what we know about those groups in northern Michigan, even though we've been studying aquatic ecosystems for years and years. We just don't know the species. So what I'd like to be able to do is for all of you guys to be able to take your samples when they come in that look like this and start turning in something that more looks like this, which, by the way, is the work of my insect class. This is one undersampled group that they worked on. This is syrphid flies, a major pollinator group that they worked on at Wilderness State Park. And we now have a much better idea of the syrphid flies that occur in the station area because of that. So thank you.
Thank you, Brian. We have time for one comment.
Yes?
Is there any information about the insect community from the Kellogg Biological Station which might serve as a point of comparison?
I would recommend they would take a lot of time surveying an MSU's collection. Yes, they've got a lot of collection stuff from the Kellogg Biological Station. However, their habitats are totally different. So we would have generally distributed stuff that would occur in both places, but we would lose all the specifics. So a lot of their aquatic species would be very different because they have totally different kinds of streams.
Okay, thanks, Brian. I just want to point out that the clock in the back is actually about 4 minutes fast. I'm going by real time, so we're not going to shortchange Chris Gough. Chris Gough from Virginia Commonwealth University. Made the trip up. Thanks, Chris.
Yeah. All right, well, thank you. Yeah, I'll try to get everybody to their coffee here shortly. Bob set me up nicely, although I have a dispute that we're only interested in a big leaf in the forest, but might be oversimplifying it a bit. But I want to actually build on some of what Bob talked about earlier, talk about forest resilience following disturbance and what we've learned, kind of give you an example of what we've learned from the FACET project. The entire FEST team has really been involved with this, so if you're interested in this work, you can talk to Peter, Knute, Luke, Bob, others who are involved, Chris Vogel. So why did we kill all those trees anyway? Well, initially, the Forest Accelerated Succession Experiment, right, FACET, as the name would suggest, we're really interested in what happens to the biogeochemistry of the system after we kill these early successional trees, as the forest advances beyond the early stages of succession. So this is, Chris, I think, maybe in 2005, which we ended up not using that approach to girdle. But in addition to advancing accelerated succession, we also created this, as Bob mentioned, really this gradient across the landscape in disturbance severity, right? And so if you look at within the large manipulated area of FACET, the 32 hectare area, you can actually see sort of this variability, right, in mortality within the large plot. So that actually presented a nice opportunity to study then what happens to the carbon cycle across a gradient of disturbance severity. And this is useful and important, we think, because biogeochemists like to study sort of these extremes. So what happens to the carbon cycle following clear-cut harvest, wind throw, catastrophic fire? That's been around. That science has been kicked around for a while. There are a lot of flux towers also kind of in background, low levels of disturbance severity, but not really in between. There's not a lot known about what happens in the middle, right? So what are the responses in between? And so FACET, we think, fills in kind of nicely this middle area of disturbance severity, as Bob alluded to. And essentially, you can think of 3 potential short-term responses of production to increasing disturbance severity. So if this is disturbance severity on this axis, and this is net ecosystem production or primary production, we might expect there's sort of this baseline production value, right? So at the low level of disturbance severity. And if we increase disturbance severity, we might expect a linear decline in production, right? And actually, this is sort of our hypothesis, right? It makes sense. You kill a certain number of trees, and you see an incremental decline in production. Another possibility is one of high sensitivity to disturbance, so a precipitous decline in response to a fairly low level of disturbance severity. And then one is of resistance. And so Bob kind of, don't forget what he said. I don't want you to remember the punchline, but this is another possibility, right? That there's high resistance until we exceed some threshold of disturbance severity, and then we see a precipitous decline in production. And so at UMBS, we have a nice, landscape of variability in disturbance severity represented. And some of that is in the FACET site, but some of it also is, the green here represents the Ameriflux tower, so the control site that's been operating for over 15 years. We have a flux tower operating, several inventory plots where we can measure NPP and NEP. So this is sort of a low disturbance severity scenario. And nearby, we've got the burn plots, which have been clear-cut, harvested, and burned within the last, a couple times really, within the last century. So this gives us kind of the high disturbance scenario. So we can ask the question, what happens to production following, or sort of in this low disturbance area background, and also following this catastrophic stand replacement disturbance? And when we look at the data, we look at net ecosystem production here in time since severe disturbance. And this is from, again, the control tower. Net ecosystem production is kind of humming along. You kill all the trees, essentially, right? You clear-cut harvest. And NEP tanks, not surprisingly, right? So it declines actually to below 0, so the ecosystem is probably emitting CO2 briefly, and then a recovery period. And we can pick those points off the curve, right, and go back to this hypothesis curve, and we can then plot sort of where the low disturbance severity scenario lies, and then also the high disturbance scenario. We get those 2 extremes, right? But we don't know what's in the middle, right? We haven't yet resolved what's here in the middle. And so that's where FACET really comes into play. So revisiting our landscape at UMBS, we add FACET into the mix here, where again, we also have a flux tower operating, several plots, inventory plots. We're measuring NPP and NEP. And again, if we revisit that landscape within the larger manipulated area, we can see that there's a lot of variability in mortality. And so Ellen used that gradient in stem mortality within, really, which ranged from 6% tree mortality within a plot to up to 69%. So a fairly broad range in moderate disturbance severity to ask the question, what happens to production? What is the response across this broader gradient? And Bob showed you this, but you don't remember this, right? It was all new to you. In fact, we did see this resistance, a response that was indicative of high resistance to moderate disturbance. And so we see, again, this is net primary production, really the change in net primary production. Think of this as increasing disturbance severity going this direction. And so this slope is essentially equal to 0, so it's not significantly declining here. So we see that there's high resistance, we hit a threshold, and a precipitous decline. So again, we find support for high resistance up to a fairly high level. You can kill 60% of the trees, and net primary production is sustained. Right? So that was somewhat surprising, and we could overlay those plots and the results there, and this is what it looks like. And so, as we like to say in our group, maybe use this a little too much, but I like it, you know. So what's going on, right? Why is this? Bob discussed why this is in part, but basically, without showing you data, we saw a very rapid canopy structural reorganization, which improved resource use efficiency. So essentially, there were fewer leaves, but the leaves were functioning at a higher level physiologically. so they were more physiologically competent. And so that occurred much more rapidly than we expected. And then the second is, and Luke's done a lot of work looking at the retention of nitrogen, Luke and others. And so that nitrogen was retained. And that was important too, because the UMBS forest is very end limited. So that retention and reallocation of nitrogen was really important to sustaining production. So what have we learned? Revisiting our hypotheses, and this is always useful to do every now and then, right? Essentially, we found a negative result. We weren't expecting, again, to see such high resistance. So we sort of thought forest production, again, would decline linearly, and that didn't happen, right? So an interesting case of sort of negative result, but a surprising result, but probably more interesting. A second hypothesis was that we expected to see production increase over time, but essentially this occurred much more rapidly than we expected. So I crossed out that long term. We were almost right there, but it was really the short term reorganizations that occurred much more rapidly than we expected. So what's next? What's the next big thing? I'm not sure. I really don't. I hope we can talk about that maybe later this afternoon, I think, and even after the coffee break. I think as a core site, we have an opportunity to think long-term, right? So that puts us kind of in a unique position. So the long-term core site status of the Ameriflux project at UMBS essentially means we don't have to write proposals every 3 years. And we think we will have sustained funding for the next decade or so. So I think it puts us in a unique position to think long-term, and that'd be something maybe to discuss later. I think we'll continue to study structure-function relationships. We've got some new toys floating around, balloons and planes and drones, and but exciting stuff, right? So I think we're still kind of pushing this. I think trying to understand structure-function relationships, and also the next stage of recovery, really, or the ecosystem may have recovered, or net primary production hasn't changed, but certainly it's a different ecosystem, right? It's a reorganized ecosystem. Synthesis and modeling will continue to be an important part of what we do, and I think there's a really good opportunity here for students, for example, who may be interested in synthesis activities, and UMBS has been very much involved as a partner and a contributor to these synthesis activities. And then I would just say, even greater interdisciplinarity moving forward. I always tell my classes when I give kind of the spiel on what we do at UMBS that we're a very broadly trained group, right? And by the way, these don't necessarily fit these people's expertise here, but the students never know, right? I mean, but the point is, we do have a lot of interdisciplinary, and I think that's really been important, right, to advancing the science that we do, and I think we have additional opportunities. This is new work certainly for me, and I look forward to learning more this summer. So if students, faculty, if you have interest in the broader experiment, think you might be able to use it, please talk to someone on the FAST team, and that's all I got.
Thanks,
Thank you.
Chris.
Yep.